Good day, everyone. Thank you all for joining us for our live webinar today entitled Advances in Determining Circulating Ceramides Level, which will be presented by our guest speaker, Federico Torta. My name is Maria Federova and I'm based at TU Dresden. This webinar will be recorded and will be uploaded on LIPID MAPS website and our YouTube channel within the next 48 hours. The webinar will be around 45 minutes, allowing around 10 minutes at the end for questions. Please use the questions and answers function in the Zoom through the presentation to ask your questions, and we will unmute you to ask your question directly to Federico at the end of the session. So now I would like to introduce our speaker. Please, Federico, can you turn your camera on? So Federica is a research assistant professor at the Singapore Lipidomics Incubator, or SLINC, and Department of Biochemistry. And he is also the director of the lipidomics unit of SINC-MAS, the Singapore National Laboratory for Mass Spectrometry. He received his PhD degree in biochemistry from the University of Parma in Italy, and his expertise includes sphingolipidomics, high-throughput technology for assessment of lipidomes in large human cohort, and study of protein-lipid complexes. So please, Federico, you can share your screen. The floor is yours, and we are looking forward.
Thank you. Okay, thank you. Thank you, Maria, for the kind introduction. And thank you all for attending this webinar. I'm really glad and grateful to have the opportunity to contribute to this series of webinars. And today, I will talk about ceramides and their measurement in human plasma by mass spectrometry, of course. So initially, I will just explain briefly what ceramides do and what is their function, and also why it is important to measure them from a clinical research point of view. Then, for most of my talk, I will illustrate how the lipidomics community put together an international trial to which I guess many of you contributed to advance the ceramide measurements. And in the last part of the talk, if I still have time, we show a recent contribution of our lab to this field by performing ultra-fast measurements of ceramides in human cohorts. Now, ceramides are a class of sphingolipids that play a role not only as structural component of membranes, but also as bioactive molecules. Ceramides contain a sphingosine backbone in blue in the figure, and fatty acid in yellow, linked by an amide bond. They are synthesized starting from acetyl-CoA and serine, and can then generate more complex sphingolipids, such as sphingomyelins, complex glycosphingolipid, or S1P, which is another potent signaling lipid that can then be degraded to exit the pathway. Now, when we have an excess of free fatty acid at the tissue level, such as in obesity, for example, what happens? They can either be stored as triglycerides, or they can be used to produce energy. But when a cell's energy needs are met and its triglyceride stores are full, then acetyl-CoA enter the ceramide biosynthesis pathway and the level of ceramides increases. And when the ceramides accumulate, they can trigger several cellular processes, including apoptosis, mitochondrial damage, inflammation, and insulin resistance. The ceramide synthesis that I just described takes place in separate cellular compartments and includes three distinct pathways. In the ER, we have the de novo biosynthesis. In lysosomes, we have the salvage pathway, which recycles the sphingosine backbone by degrading the complex sphingolipids. And then we also have the sphingomyelin hydrolysis, which happens mainly in the plasma membrane. The disruption of this ceramide metabolism affects their cellular levels locally, but also in circulation. And in the circulation, sphingolipids are present in lipoproteins, including chylomicrons. And ceramides, in particular, are high in VLDL and LDL, but are also present in the other lipoproteins. Now, recently, with improvement of isolation of extracellular vesicles, we know that they are also enriched in sphingolipids, so ceramide might also circulate in extracellular vesicles. And as we said before, ceramides can accumulate in individuals with obesity or dyslipidemia, and they will affect cellular processing, starting the tissue dysfunction that can then cause diabetes and heart disease, for example. And it might sound a bit counterintuitive, but there is now evidence that ceramides that accumulate under these conditions might actually help cells handling the excess of fatty acids. And they do that through facilitating their import through induction of CD36 on the plasma membrane. They can activate genes for the TG synthesis, and they reduce uptake of glucose and the mitochondrial activity so that they can favor the use of fatty acid as a source of energy. So as ceramides accumulate in these conditions that I described, they have been attracting attention as biomarkers for cardiometabolic disorders. This was very well summarized and discussed in this essay written by Scott Summers, explaining how there are analogies between cholesterol and ceramides as predictors of cardiovascular events. And although ceramides are present at a level that is 1,000 times lower than cholesterol, they can now measure quite accurately by mass specs. And we found that circulating ceramide levels correlate strongly with future cardiovascular events, such as myocardial infarction and stroke. The question now could be if this correlation are strong enough to justify their use as a diagnostic tool, for example, and also if the medical community is prepared to make clinical recommendations based on circulating ceramide levels. And eventually, which are the possible interventions that can modulate the ceramide levels. So all of this is also discussed in the in a nice feature article published in Science last year and written by Mitch Leslie. So if you have time, try to give a look because it's a very interesting article. But how do we know that the ceramides are risk markers for cardiovascular diseases? Most of the initial work was done by Zora Biosciences in Finland. It started with a purely untargeted lipidomic analysis that revealed how certain ceramides were associated with the risk of cardiovascular events. And then the results were validated on other cohorts, and the list of lipid markers was reduced to 4 ceramides, and then 3 of their ratios that were used to estimate a risk score for SERPs. After that, many other research groups were able to reproduce these results. And also another score called SERP2 that incorporates additional phosphatidylcholines, in particular 3 of them, 2 containing polyunsaturated fatty acids, and a saturated one was introduced into practice. Now, these models have been validated in several large-scale studies for both primary and secondary CVD risk stratification, and were adopted by the Mayo Clinic in 2016. So now Mayo offers these panels. And more recently, I think since a couple of years ago, even by Quest Diagnostics. So what is also important to note is that the ceramide scores, these ones I just described, are a complementary tool. We can see it as a complementary tool in CVD risk prediction when compared to other lipid markers, for example, LDL cholesterol, because these groups found out that they are quite independent markers. So that means that integrating the search scores with other risk predictors or other models, such as, for example, high sensitivity troponin assays, improve the prognostic potential of these tests. And yeah, let's keep in mind that all this was done by LC-MS/MS, so a technique that is very close to what we do, and how then this translated into the ring trial we'll talk about in few seconds. Okay, so a few years ago, some of us met here in Singapore and others connected online, and we really came together as a community of lipidomics researchers to discuss how we could improve our quantitative analysis and make them more reproducible between different labs. And back then we agreed that the measurement of ceramides could be a good first candidate for this kind of optimization for several reasons. So first of all, the clinical relevance that I just described. Then the stability of ceramides in plasma, which is quite high, their detection levels, and the fact that there are quite a few established methods for their measurement. And I really have to thank primarily Marcus that put a lot of efforts and support into this initiative. And then together with my other colleagues, Niels, Bo, Kim, and Robert, we coordinated an international ring trial that in the end involved 34 labs all over the world. And I would also like to take this chance to thank again all the participants. You might be able to see their or your names here. They have been great and very collaborative to support all the phases of the study. So it was really a pleasure to work with all of you. And of course, we also received a very essential support from both Avanti and NIST that provided the internal standard and the reference materials for all the participants. So thanks to them as well. Now, sorry. What were we asked to measure to the participants of this study? So we asked to measure the 4 ceramides included in the CER score, of course, and their corresponding deuterated standards. What was received by each participant included 4 plasma reference materials. The choice of the NIST SRM 1950 was quite easy, and then was due to its commercial accessibility. And indeed, this has been used in many lipidomic and metabolomic studies already. While the other 3 reference materials used in this ring trial allow us to test different matrices which might be useful in future clinical research. As ceramides are implicated in cardiometabolic diseases, we included pooled samples from populations with high TAG and diabetes, for example. And then because ethnicity is another factor that can affect plasma sphingolipid levels, we also analyzed the young African-American blood plasma as one of the reference materials. The participants also received 1 vial with pre-mixed internal standards labeled, and 1 vial with non-labeled standards to build the calibration curve. And we then also sent an SOP based on a validated published method. So this SOP included quite detailed step-by-step procedure for the ceramide extraction from plasma, for the preparation of the external calibration curves, and for the use of the MRM-based CMSMS method. There was also a template to design the sample analytical sequence and to report data at the end in a standardized format to make the final analysis easier for us. And this was included as an Excel document. But we also asked the labs to measure ceramides with their own preferred method for comparison. So labs could decide if using only the SOP that we standardize or their own method or both. Now, the major aims of this study included evaluating the concordance between labs and platforms, to highlight the technical issues that could contribute to variability in case and the outliers, to report absolute concentrations of the 4 circulating ceramides in all the 4 standard reference materials with both single and multi-point calibrations, and hopefully also to lay some foundations for future measurements and future determination of lipidomic reference intervals for, in this case, ceramides in diverse human populations. So now finally I can show you some results. We received a total of 39 data sets. The majority used the standard methods, the SOP, but quite a good number used also a different preferred method, which was great for our comparisons. While the SOP method only included reverse phase separation and the triple, quadruple, or Q-trap-based analysis, the preferred method were also performed using either reverse phase or flow injection, in one case supercritical fluid chromatography, nano or Kapel-C, and also high resolution MS. So we try to compare all these results, but of course we have to be fair and say that the number of labs contributing, for example, high resolution data or separation different from reverse phase was quite small for a proper comparison between platforms. These are all the data obtained for the SRM 1950. Each panel is one ceramide. In red, we have the labs that use the SOP, and in blue, the others. We perform outlier filtering for the final calculations, but here you can see represented all the results. And the outlier filtering was applied at the threshold, which was a bit more than 2 standard deviations. What we can say about the data is that we got a very good intra-lab reproducibility, which was expected, but we also observe a good concordance between labs. Indeed, the CEV were all below 15% for all the 4 ceramides. The concordance of the results between the participants was not affected by the use of the SOP or other procedures, as the values generated from these 2 groups of methods were not significantly different. And now, again, because of the difference in the number of contribution that we got from low versus high-res instrument, we did not do any statistics for this comparison, although we did not find significant differences also between these 2 sets of results. We then compared the quantitation by using single point or an external calibration curve, as both methods are currently part of our lipidomic workflows. So we obtained comparable interlab CVs for concentrations calculated using the 2 methods, and you can see that in the red box. But in terms of concentration values, and you can see that from the graphs on the top, while we observe the same values for ceramide 24.0 and ceramide 24.1 with both calibration methods, we observe different ones for ceramide 16.0 and ceramide 18.0. And for example, in case of ceramide 16, we saw a 1.4-fold higher concentration for single point when compared to the multi-point calibration. And the opposite was true for ceramide 18.0. Now, we do not have a good explanation for this discrepancy other than possible disagreement in the amount between the labeled and the unlabeled standards in case of these two ceramides, 16 and 18, because we also observe it in the calibration curves that we prepared in the bovine serum albumin solution. Then, when we compare the different reference material to the 1950, so the 3 additional reference material, we observe a clear trend for all the ceramide species with the highest concentrations present in the high TG plasma, and with the lowest values in the young African American plasma. As it was already previously reported by other studies, they used the same set of samples, and suggesting that probably in the future we will have to consider stratified reference intervals for applications in clinical testing. The one we used here is a package of 3 pooled human plasma materials, as I said, from diabetic, high TG, and young African American. But and this, I think, is a good reference for this kind of studies. But we also have to say that unlike the 1950, which was pulled from 100 healthy individuals, these are prepared from a smaller number of individuals, usually between 11 and 16, between all of them. And there are not many values certified or referenced like it is for the 1950. Although I think we really have to appreciate the efforts that NIST is putting in supporting the lipidomic and the metabolomic researchers and generating this new set of samples that will be very important for estimating the effect of different type of matrices, especially for this kind of applications. This graph shows clearly the importance of using authentic standards for quantitation. This is, I think, a very important point that emerged from our study. So it was, of course, a choice from the beginning that, to overcome complications such as different extraction efficiencies or ionization and fragmentation properties and dependence of these properties in the chain length. And then, of course, lack of coelution, we use authentic standards from the beginning. So, but to evaluate the relevance of this choice we made, we normalize the peak areas for each endogenous ceramide species from each participant. You see them all represented here with either the authentic standard, which is, in this case, the corresponding stable isotope, or with one of the other 3 available label standards that we use to normalize the other 3 ceramide. So in this figure, you can see a simplified representation of our results with only 2 standards, but they all behave in the same way. And so in one case, we estimated the concentration and the CVs between the labs of ceramide 16.0 by using the authentic standard for 16.0 or the deuterated standard for 24.0. And on the right side, we did the opposite. So what did we learn from this? Besides the mean concentration difference, what you can really notice here is the variabilities that were really significantly affected by the type of the internal standard that we use to normalize. And for example, in the figure on the right side, right, resulted in interlab CV of 10% when we use the identic standard versus about 60% CV when using another one. Now, of course, we can say that in this case, the structure of the two standards is quite different in terms of chain length and mainly in terms of chain length. But we look also at all the other standards. We compare 16 with 18, 24.1 with 24.0, and we obtain very similar results. Now, the reasons for these results is not clear, totally clear. So we could consider, for example, matrix effects and different efficiency in source in terms of ionization between different structure, different fragmentation efficiencies for the different analytes. All these can have a relevant effect. And we thought that, for example, some of this variability could be minimized by, for example, using HeLiC or SFC separations or flow injection, where we have coelution between the analyte and the standards, right? So that we can have a better co-ionization and due to coelution, of course. But so we look at the specific data sets that use these techniques, but of course, because the number of these data sets in our study was very small, we could not reach a definitive conclusion and say that by using this separation technique or without even separation could solve all these issues, because in some of them the issues were solved, but in others it wasn't. So we need more data to test this hypothesis. Now I would like to talk about harmonization, because although I showed you, we got already quite concordant results between the participants. We also look into how to improve them even more by harmonization between different platforms, because all our labs use different platforms and some also different methods. And this is, and the harmonization procedures is, of course, also very useful. For example, when we run multi-center studies or longitudinal ones. Now, the idea that we explored previously and also others have published is also suggested by current procedures in clinical chemistry, where you can add an external reference material to our sequence of samples, analyze it together with the samples, and then normalize the concentration of each analyte using the ratio between the concentration of the analyte in the sample and in that reference material. And how did we do it here? So we basically used NIST SRM 1950 as an external reference. And this is also because it's commercially available and one of the most popular references for human plasma studies. So we harmonized the data between all the participants by doing this. So we took the 4 ceramides concentration values obtained from the other 3 different reference materials, the high ATG, the diabetic, and the young African American one, by each lab. And we normalize them by the mean concentration of the same corresponding ceramides that we obtain that was obtained by the same lab for the SRM 1950, according to the formula that I have represented on top of the graphs. So this gives us a unitless ratio that then is rescaled by multiplying by the concentration that the consensus concentration that we assigned to the SRM 1950 in this study. So we basically went back to concentration values. Now, after this recalibration, and then you can see in the figures that I reported below here for each ceramide, the values that we obtain before recalibration and after recalibration with the SRM 1950. So the interlab CV decreased for all the reference materials. Here I just represented the diabetic plasma. And what you can see also is the number of outliers significantly reduced. And the decrease in the interlab CVs was actually reduced by 5-fold for all the 4 ceramide species. And it's not only the effect on the CV that we have to highlight, but also that the mean consensus value for that specific sample is not so significantly affected by this recalibration. But we can really say that the main effect is on the comparability between measurements that we generated with different methods or with different instruments and different procedures. So this, I think, it's something that has to be tested further with many more studies and many more different plasma matrices. But I think it seems to be quite effective procedure to really harmonize results between many different labs. Now, as I mentioned earlier, we also tried to understand what could be some of the technical reasons behind the outliers. So we didn't have many, but we went back to the raw data, tried to understand what could have happened there. In few cases, we could not figure out what really happened, but in others, we obtained some interesting information that we could make use of for future experiments. For example, by looking at the raw intensity values, we realized that one of the outliers probably spiked in 3 times more of internal standard. And so, of course, all the concentration values were very low compared to the other labs. Another, that happened once, but another more recurrent error was due to copy-paste errors in Excel. Basically, columns were swapped, or the sequence of the samples in the calibration curve, for example, was shifted. And this, of course, affected all the final calculations. So this, of course, means that we have to reach a higher degree of automation, of course, not only in sample prep and the analytical steps, also in the data analysis as much as we can. And then one last factor that I would like to highlight was that some outliers were affected by the contribution of the signals from isobars, where, for example, when the measurement were based on MS1 measurement, because in that case, no correction was applied, and so the signal of the isobars was summed up to the signal of the ceramide. Now, to conclude this part of the talk, we can say that to obtain concordant data that we obtained, the method standardization helps, so that by sending out SOPs, but is not fundamental, because even labs using their own preferred method were completely aligned with the standardized one. Then another thing is that the authentic label standard and harmonization procedure with the reference material reduced significantly variability between measurements. And that finally the reference values generated in this study can be used as a quantitative benchmark for future measurements. Now, if I still have some time, but please let me know if I'm going too long, and then I will stop anytime. I wanted to, for this last part, I would like to show a contribution to this field also from our own lab. So what I showed so far was in a few commercial samples, but how can we improve analysis of ceramides in big human cohorts in the order of thousands of samples? For example, if let's say we want to generate reference values in population or find genetic association with the levels. So together with Whiting, one of our industrial PhD students with Agilent, we thought we had to reduce significantly the sample preparation and analysis time if we wanted to do these big studies more efficiently. So we integrated two solutions, a robotic system for sample prep and a rapid fire system connected to a triple quad for analysis. Now, the rapid fire is an online SP that operates at very high flow rates and can allow us to analyze samples in a few seconds. So Whiting optimized the conditions for binding of the 4 ceramides to the SP cartridge and also for the elution into the mass spec in 15 seconds per sample. So that allowed us to analyze a cohort of 2,000 samples in one day with a very low CV. So now let me show you some data. To test our method, we measure first the same set of samples with using the same SOP of the ring trial and using a CMSMS and with our new method on the rapid fire. So we've been working for a while with Tony Futterman and this ceramide synthase 2 null mice model that we thought is an ideal model for this test. And in this model, the ceramide synthase 2 does not synthesize very long-chain ceramides. And while there is an increase, a compensatory increase in long-chain ones in plasma. So when we compare the mouse plasma with both methods, LC and rapid fire, we observe identical results in both the wild type and null mice. So this was quite encouraging. And what we did next, we did the same approach. We compared the results obtained with both CMS and rapid fire when analyzing the 4 ceramides in 100 different human plasma samples from the hospital. And as you can see from the regression plots and the Pearson correlation, these measurements were very reproducible between the two techniques, even when we consider a higher matrix variability between individuals. Now, how do we use this approach in real studies? We did this in diabetic in a type 2 diabetes study. So cardiovascular disease is the leading cause of death in patients with diabetes. But at least to our knowledge, the use of CERT1 score, the one I described before, for prediction of mortality has not been systematically assessed in patients with type 2 diabetes. And also the association between the CERT1 score and the risk of non-cardiovascular death has not been reported in patients with and without diabetes. So together with our collaborators at KTPH Hospital here in Singapore, we use these prospective studies in which we try to assess whether the baseline CERT1 score may predict all-cause and cause-specific mortality beyond the clinical risk factor in individuals with diabetes. So we analyzed around 2,000 samples from this cohort with 10% mortality rate. We used the rapid fire method to measure the ceramide concentration. And then at the end, we calculated the CERT1 score to stratify into risk groups. And as you can see from this figure, patients in the higher score risk based on CERT1 definitely show a significantly higher proportion of all-cause mortality. From this table, you can then appreciate that this is based on Cox regression, and the participants with the scores in the intermediate and high categories that we showed before had respectively 1.44 and 2.46-fold increased risk for all-cause death as compared to those in the lower risk category. And we also perform adjustment for demographic variables and cardiometabolic risk, but this did not change the association, and the further adjustments for EGFR and then albuminuria attenuated the magnitude of the association, but still the association between the high ceramide risk score and all-cause death remained statistically significant. Adjustment for medication changed only minimally the strength of this association. And here we also look then into cardiovascular and non-cardiovascular death associations, and compared to the one in low risk category, the participants with the SIRT1 score in the high risk category have significantly higher risk for both cardiovascular and non-cardiovascular death. And then finally, when we added the SIRT1 on top of two different risk scores that are normally used for this patient, it significantly improved the prediction of 10-year risk of all-cause death. So this tells us that measuring SIRT1 could improve prediction for this kind of cohorts. Okay, so we reached the end of the presentation, and I would like to acknowledge the people in our lab that contributed to the results I showed, which are Snea, who's now working in the US, and Dimei, and Waikin and Jinkai for the ultrafast measurements, and of course our collaborators at KTPH Hospital that collected all the samples and guided our analysis for the diabetic cohort. And then I would like to thank Marcus for his support with everything that has been done in these studies. And back to the ring trial, there were key people at NIST, Christina, Catrice, and Tracy, and also in Avanti, Lisa and Tucker, that gave an essential contribution to all the operation. And again, thanks to all the participants in the ring trial, as it was really a pleasure to work with them, and finger crossed for our paper that is still under revision. And thank you all for the attention, and I'll be happy to take any question.
Thank you very much, Federico, for the great talk. So it's there is a question from Mangala Prior. So I'm going to perform untargeted lipidomics in diet-induced animal model for untargeted lipidomics, whether to prefer DIA or DDA.
Yeah, that's a good question, and maybe you, Maria, are more expert than me in that. We do perform quite a lot of targeted analysis here, and only now moving more towards untargeted, where DDA and DIA are the preferred methods, right? So I guess it also depends on if you trust your data analysis part, right, of the experiments. DIA is an interesting approach, maybe more robust in proteomics nowadays. With lipidomics, I think we are still playing around with it, especially with the data analysis part, because it's not trivial, right? Because you're going to fragment everything. But of course, there are software like MS-Bial that now probably perform quite well also with that. So I think it's really up to the expertise that you have established in the lab, what to choose, probably. So maybe, Maria, you have more insights about that.
I do agree with you completely. I think the main point is DDA versus DIA is the availability of the tools to actually deal with the data, right? So the convolute via data, I think it's still quite problematic. So. The other question also here is about whether it is mandatory to use pooled quality control samples to check data integrity.
Yes, so we always do that in all our studies. We include pooled quality controls. Of course, then is up to you, and then basically the size of the study to decide if you want to pull all the samples in your cohort, or maybe a subset. If you can, of course, it's always better to pull all of them. But and we realized that in the end, 30% of the samples that we run are made by QC. So we have now quite different types of QC. Of course, it's not only the pool samples, but that I think is quite. I mean, there's nothing mandatory, right, in our approaches. We can be flexible if things make sense, but I would strongly suggest to always include it.
Absolutely. I think in my lab, we learned a lot from you, Federico, about all the necessary QCs we are using now as well. So the next question is from Al Merrill.
Federico, very nice presentation, and it's amazing to see how far advanced this has gotten in the last few years. The only question that I wanted to propose was, since you have so many different tools and the interesting samples, do you all periodically go through and do a scan across potential ceramide subspecies of the kinds that are not normally thought of, double bond containing fatty acids and sphingoid-based backbones, chain length variants, that sort of thing, both known mammalian ones and perhaps also some of the ones from dietary sources, to begin to get and the 1-deoxy molecules, of course, are especially in diabetes, something to think about. So that this is sort of a two-dimensional question. One is getting better and better and better with the major species that people look at, but also expanding into the realm of what would more or less be considered the horizon of a fully comprehensive ceramidomic analysis.
Yeah. Hi, Alfred, and nice to hear from you. Of course, all these things were just possible because all the things you did before us. So yeah, but as you know, we have this kind of two different approaches, as you just described. One is very targeted and more, I guess, focusing on the methodological part, especially when we want to go into clinical, right? So we have to really focus on robustness and reproducibility and precision and all these kind of things, where it's easier to start focusing maybe on a small number of lipids. And the ceramides was a great case because there's 4 of them that have been already used in the clinic, and was kind of ideal system to optimize in the community. And we learn from that. And indeed, now the next ring trial, which is already basically finished, included a much bigger panel, but with same kind of purpose. So we are learning more and more while we do that. And in our lab, the other side of the sphingolipid analysis goes exactly in the direction that you mentioned, where we either do untargeted or even a very wide targeted approach. Now, I think we include about 400 different sphingolipids in our methods. So of course you have quite a long gradient over space, so we can separate all the species that you mentioned, and in that case we really look into subclasses that differ by the long chain base length and saturation, by the modification and the desaturation, saturation degrees, and sugar moieties. So it's a very interesting part that we develop in parallel, of course, and where maybe at this stage relative quantitation, or let's say semi-quantitative approaches are still fine, and that where we can use mouse models like with Tony, for example, exploring many different tissues, but also moving this kind of wide approach into plasma now in these different human cohorts that we collected over the years. Of course, these are much longer approaches if we want to be so detailed, but there's a lot of interest, right? Because as you know, people didn't really look into this kind of species much. Of course, now there's more and more on the dioxy. Sphingolipids are very interesting, and then we also find in our diabetic cohorts. but there's probably much more than that we still have to explore.
I would like also to ask a question, Federico, since you measured it in different cohorts, what about the sex dimorphism? Do you see the differences in CERT score between male and female?
So the measurements that we did, that I just showed, we didn't really see significant differences. And also there's a paper from Mayo where they studied the biological variation of ceramides in healthy individuals, where for that specific cohort, now it was a very small cohort, like 25 people, but because when you measure biological variation, you have to longitudinally sample them for months, right? So that's why of this limitation. And they didn't find for these 4 ceramides specifically significant difference. But there are other studies, for example, I think some maybe from LIPID MAPS also, where they show a variability of sphingolipids between sex, but also due to the age. So this difference might change with time between men and women. So, and what we know is now we're not looking at this specific species that we mention a lot of times today, but when we consider a D18-2 sphingoid base, there where we see big differences between male and females, right? And that I think is linked to probably FATS3 variations, and that was reported not only in ceramides, but of course all the other sphingolipids that include D18-2, many different studies. Peter Michael reported several times, and we also observed it. So sphingomyelins is very clear, for example.
And if you look on the CERT score on the sphingomyelins?
Yeah, these are, of course, the one that we show are these two CERT scores, but there's many scores out there that, researchers develop over the years with other cohorts, and some of them include sphingomyelins, for example. So these ones, we keep mentioning these because they are already in the clinic, right? So it's easier to make a case. But there are others developed by Scott Summers and other researchers as well in the US, where, in their case, maybe in their cohort, it was better to include a couple of sphingomyelins on top of the ceramides and maybe not all the 4 of them or all the ratios. And they gave them better risk prediction. So these are, you can find them in literature, definitely.
There is also a question from Anna, which is of course also a very interesting one.
Federico, firstly, thank you. It's been a great talk, and I'll echo Al's comments about the progress in the field. This is fantastic. And since Al has stolen my question about other subspecies and the importance of other ceramides, I wanted to ask you, we all measure plasma ceramides, and we say that they're a sign of danger of systemic health and whatever. Working with animal models, do you have any insight to share with us about the origin of the different plasma ceramides? So we blame the liver for everything, or can we start understanding how, for example, the adipose tissue or inflamed adipose tissue or other tissues in the body may be reflected in the plasma ceramide profiles?
Yes, very good question. And I think we should start working on that, and because it's still a big question in the field. So I guess I just have to give answers to this, and because again, I maybe there are people more expert than me in this, but one thing is mouse is of course a very good model to study sphingolipids, but there's also differences, right? Thorsten Hornemann showed that, for example, if we want to look at different launching bases, the differences might be really significant. For example, for D16 launching bases might be quite different, the situation there between human and mouse models. And then the second thing, which is more related to what you were saying, and yes, so the lipids in plasma basically is a mixed contribution from several organs, right, and tissues, as you just said, and we don't know yet probably in which percentage they come from other tissue than liver. And you mentioned adipose tissue, but I guess there's also quite a good contribution from the gut.
Yes.
You know, when we are now actually doing that, so we are collecting all the different tissues and then and the plasma, of course, from the same mouse, and then inducing perturbation and see how the perturbation in one tissue is reflected in the plasma or is not reflected in the plasma. So we can maybe estimate which tissue has a major effect on that. And I guess because it's quite a complex study, you know, collecting all kind of tissues in sufficient numbers and analyze them. But I think that this is one of the big questions that we have, and there's probably several people working on that, and I think we are going to focus more on that, definitely.
Good luck. We just need this information, Federico. So I hope you can give us
Yes,
some
all
markers,
these,
tissue-specific markers.
yeah, all these atlases, lipid atlases in different tissues, I think will be useful
Will
for
be
this kind of stuff.
fantastic. The best of luck. Thanks again.
Thank you.
So, but I guess you can reach Federico if you would like to know more. He shows here on the screen his email. So thank you very much, Federico, for the great talk. And thank you,
Thank you for having me.
Thank you, everyone, for joining. So this concludes this month's webinar. Thank you all very much and have a very good day. Bye-bye.
Bye-bye.